Okay, thanks. Now we're going to go into part two of this interview. So part one, Dr. Kim focused specifically on the Celemics NGS target enrichment product or business. And in part two here, he's going to describe a new product they have or technology they have called BTSeq, which is focused on allowing people to quickly and accurately sequence small genomes or large fragments of DNA. So this is something that could replace, for example, primer walking, which is currently done by Sanger sequencing, and it may replace other applications where you typically would use Sanger sequencing. In addition, he will also explain and introduce a little bit about their immune repertoire business, which is focused at the pharma biotech industry. They have customers there who use this service, and that's used specifically to sequence antibody fragments, so an antibody discovery. So hopefully you enjoy it, and let's get on and watch and listen to part two with Dr. Hyoki Kim, who is the co-CEO of Celemics, based in Seoul, Korea. Thanks. Bye. Let's move on to something new that I've seen recently, which, I know Korea was very quick in being able to analyze and in some way control the COVID-19 infections. And I saw a small presentation recently from Celemics on a technology called BTSeq, which you offer as a service. And my understanding was that has the potential to replace Sanger capillary sequencing. Could you explain and enlighten me on what BTSeq is and how it could replace Sanger sequencing?
Okay, so the BTSeq is a abbreviation of the barcode tagged sequencing, and second, the coronavirus analysis. I just share a little bit of the background of the coronavirus. The very early moment after the occurring the coronavirus issues, and then we got a call from Korea CDC very late night, 11 p.m. and then they requested us to visit their headquarters, CDC headquarters. It's the 100 miles away from our presence. So in the next early morning, so yeah, we need to go there because customer asked us to visit them. So we visit Korea CDC, and then they requested us to analyze the first patients of the coronavirus patients in Korea, first patients, and they succeeded in the collecting samples of the mixed state, and not only just the mixed state, but also they succeeded in the proliferating of the viral sequences. So we received that samples and delivered the whole genome sequencing data just about 21 hours. Usually it takes about a few days, or if you use Sanger sequencing, then you would probably take a few weeks to sequence all the 30 kilobases long whole sequence. It's a pretty fast. The reason why Korea CDC called us is that actually we have a collaboration with different project with Korea CDC, so they knew our technology, and it's a fast and accurate. So,
and this is a BTSeq, is it?
It's a BTSeq
technology you used. So specific, can you give any more details on that as to how it works? And I mean, obviously the problem it solves is it's very quick and it's very accurate in terms of sequencing, but to get a 30 kb accurate sequence in 21 hours, I guess that's unusual, isn't it?
It's an usually the currently available products and then methodologies available in the market usually takes much longer than that. Usually it takes a few days to complete the sequencing and analysis. So what we developed for BTSeq is we developed a new enzyme and assay protocols. Actually, we developed the enzymes and we product our enzyme in our company. So and then we configured a new way of the sample preparation. So usually it takes more than that, but very minimal hands-on time, and then you just mix it and then rest it for the 2 hours. Then the library preparation is almost done. So then you can move on to the next stage. So the reagents and then assay system is the new thing. And then second is that we developed a new the algorithm, the analysis algorithm. So basically, it's very hard to sequence 20 kilobase or 30 kilobase long sequence in just a using just a one read. It's very hard to do that. But we use a short read, but we can configure to make a very long read in a very short time frame. So we developed that the de novo assembly algorithm. So that cuts the many of the time. And also the third, one of the reason why we can reduce costs actually is that we developed also a new barcode system that the very distinct, a unique barcode system. And then, for example, usually let's say small samples, small target size samples. If you analyze small samples, then you might pull over thousands of the samples in one MiSeq run if you do really well.
Right.
But it's very hard to tag. Then it means you need to tag different sample index in each of the samples. But usually, the most of the available products, so of the number of indexes, different indexes are 384, I guess. Most of the, or even though they don't exceed 700 around.
Okay.
So, but we developed a new way of the sample indexing technology. The index, it's a not only just a different sequence itself, but also the composition of the bases are also important, because if it's too similar, then you might have the very poor sequencing quality. So we try to develop and validate many of the indexes. Like we have over 9,000 distinct indexes, and then we developed a very efficient way to incorporate many of the samples with many of the indexes, and so we can put as many as possible in one NGS run. So it can reduce our costs. So so that's what we did for BTSeq. But the reason why we developed the BTSeq is that we actually would like to change the Sanger sequencing. Sanger's NGS technology is popular, but there are so many customers who use Sanger sequencing. And then we see that the market and then customer use Sanger sequencing because their NGS is available. And then there are a few reasons for that. The first, the turnaround time. Is a turnaround time in Sanger sequencing. You just put your samples in the Dropbox and you will get the result in the next day. So it's very fast. And then second, the NGS is cost effective and a very efficient way to sequence a large target size, large target. such as tens of genes of the exons or hundreds of the genes of the exons, even whole exomes or whole genome. NGS is a very efficient way. But let's say the only thing you would need is only a small fraction of the exons or very limited spots. Then you would need, you don't necessarily to do the normal NGS because you don't, it's not necessary. And then the setup cost of the NGS is much higher than Sanger sequencing. We received the market price of the Sanger sequencing is at around $3 or $4, and then maybe it's less than that. So it's very cheap for the one reaction of sequencing. So, and then also NGS usually returns very short read. The read length is limited, but Sanger sequencing can sequence a little bit longer than that. So those kind of needs keep customers use Sanger sequencing even though NGS is available. Yeah. And then we thought that the we also hear many of the customer voice who use Sanger sequencing is that the Sanger sequencing is advantageous in some way, but it has very limited limitations, such as when you would like to do the long sequencing, de novo sequencing, you need to do the primer walking. Yeah. And primer walking is you need to sequence, and then it's a long. So Sanger sequencing can sequence long, but it's not that long. So you need to sequence many times. But to do the sequencing, you need to have a sequencing primer. But if you don't know the sequencing primer, then you sequence and analyze, and you design the primer again, and then you synthesize your primer again, and you do the sequencing again, then you need to know the primers. So it's a repeated cycles of the primer design, synthesis, and Sanger sequencing. So it takes a long time. It's very, it costs a lot, we think. Yeah. So that's one limitations. And the other is that the Sanger sequencing is a gold standard. That's a that's well-known technology. So it's a gold standard. But actually, what researchers experience is that even though it's a gold standard, but the service providers, the Sanger sequencing service provider, they compete each other as well. So we heard that they dilute the reagents, and then so the sequencing quality is poor in some applications, and then also there are many rerun cases. So if the sequencing is a failure, then they need to sequence again. And then that can be due to the reagent problem, but also they are prone to the complex structure of the DNA, and then maybe the primer is not very specific. So because of due to the many reasons, they have many rerun cases. So they are. Sanger sequencing is advantageous, but they have also many limitations. So we would like to overcome all of those limitations of the Sanger sequencing, but keeping the advantage of the Sanger sequencing. So we developed a BTSeq to solve that problem. So that's why we developed one.
So the advantage is mainly, as I understand it, with Sanger sequencing, it has the far better accuracy normally on a run than NGS, and obviously longer individual runs, and it's relatively inexpensive per sample. So just to summarize on BTSeq, you would maintain, or BTSeq maintains the accuracy of Sanger sequencing, and if you have the sample volume, it's competitive on price.
Right.
Right, but you get far more data, up to 20, 30 kilobase data per run.
Right.
Okay.
And then also it's a very it's fast. You don't have to have a primer design and synthesis and then sequencing, and then it's not necessary to do the primer working cycles.
Right.
So even though we are requested to analyze over 10 kilobases or 20 kilobases, our turnaround time for the sequencing of the entire 10 kb or 20 kb is 2 or 3 days. So but usually you need to wait much longer than that
On Sanger,
on Sanger, right?
Okay, there was just one other thing you mentioned at the start, Hyoki, and maybe you could just say a couple of sentences on this, but it was immune repertoire. That I understand is related to, well, you explain instead of me explain.
Okay. Okay. So our immune repertoire analysis solution group, it's a product group, and we have 3 products in that product group. So the first is the TrueRepertoire, and the second is the immune profiling analysis service, and then third is a MRD kit. The MRD stands for the minimal residual disease. So briefly, the first TrueRepertoire is a solution that can analyze it's a very high throughput version of the cloning and sequencing analysis. So for example, we have many customers in antibody development in pharmaceutical industry that one of the routine work they do is to make libraries and then do the bindings and then see and they're finding the clones, the hits, and then they sequence what is the sequence. So that cycle is a biopanning cycles, and then in that biopanning cycles, they need to, what they really need for them to have is as many as right clones as possible. But it's very hard to have that clone because of the limited throughput of the conventional cloning and Sanger sequencing. So we have our team that one of our team member who was actually our customer sectors, pharmaceutical industry. She developed antibodies for like 20 years. So she joined our team, and then she told me that when she was in a pharmaceutical company and developed drug antibodies, then it's a physical limitation that one researcher can handle a few clones. So that's a physical limitation, time and money and everything. So we our TrueRepertoire it's a very high throughput version, so that not only just the cost and speed, but also actually it can find rare clones, but it's a positive clones.
Right.
Much effectively. So that's a TrueRepertoire. And the second is the immune profiling. Immune profiling is a bulk analysis of the features. TrueRepertoire is a clonal sequencing, a clonal selection, and then we sequence the clones. But immune profiling is a analysis of the bulk feature, like a 10 to the 9th to the 10 to the 11th, or even in the 10 to the 12th of the complex library. So we developed an assay to retrieve a very uniform of the repertoire sites in the sample. That's the first thing. And the second, we developed a complex bioinformatics algorithm to make more accessible to our customers. It's a large big data. So you need to, but you need to find the meaningful information. So we developed that service with one of uh, um, um, the leading groups in Korea.
Right.
And then we have publications. And then third, the MRD kits is actually we are developing it. The TrueRepertoire and immune profiling services are ready to provide, but MRD, it's an R&D phase, and it's a when patients have leukemia, then you might have the treatment, and then your cancer cells will decrease, but in some time point, those cells can relapse. Then you need to monitor how those the treatment goes well, and how those relapse goes on. So
Okay. Yeah.
yeah, so that's the MRD kit.
Okay. Now your company, or as a business, you've been very successful in Korea. I believe you're a market leader in Korea, and you're now expanding worldwide, and that's kind of how we met each other. What are the challenges you have as you expand from Korea into other markets?
Okay, so actually, one of the challenge is the physical limitation, definitely. So we try to be a global company, but for now, we cannot directly develop our customers in everywhere in the world by ourselves. So so we need partners. So actually, that's one thing that we need to find to the right partner for the specific region. That's one thing. The second, some of the barrier that we need to overcome, and then we are focusing very much, is to overcome the brand recognition. So our peer companies, they are most of our companies are come from the United States companies, and they are big companies, and they are very well-known companies. But compared to that companies, we are not that known in the market. So we need to make ourselves more present in the market, and then we need to accumulate our trust of our products and the reliability of the products and company in different countries. So we are very much focused on to overcome that barrier.
Okay, well, you have I know from experience of talking to others, you have certainly on the target enrichment side of things, you have something very unique and special. And it sounds like with the other products and services you're developing through BTSeq, especially, that sounds like it's going to be very exciting when everybody has access to that. And also in the pharmaceutical industry, in the hunt for new antibodies with TrueRepertoire, must be a lot of opportunities for you. Hyoki, we're coming to the end of this interview. One thing is, if anybody wants to know specifically any information, is the easiest thing just to go to your website? Is that the best place for information?
Right. You can visit our website, and then look around. And if you have any questions, then feel free to ask us. You will have the email address below the website. And then also, actually, we are developing new website, and then we iteratively release in a very short time frame.
Okay, and that's Celemics.com.
Right.
The website address. Okay.
Sorry, I don't know that one.
Oh, that's my smart speaker.
Okay.
But I will. I'll add that at the end of the presentation. And with that, I'd like to thank you very much for your time. It was very informative, and I really enjoyed the discussion. Thank you very much.
It's my pleasure. It's my pleasure, and thank you for having me here. Thank you.
Thanks, Hyoki. Bye.
Bye.